Hello and welcome to the first segment of the lecture material on muscles. There will be 3 segments on muscle. This is the first segment and we will go from slide number 1 to slide 66. So as usual, here's a list of topics that we'll cover. And in segment 1, we'll start off here at the beginning with a brief introduction to action potentials. And we'll go all the way through how muscle cells and muscles contract and relax. And in segment 2, we'll pick up with muscle metabolism. Muscle cells are electrically excitable along with neurons. And what that means is muscle cells and neurons are able to generate and conduct action potentials. Action potentials are electric currents, physiological electric currents, and they're caused by the rapid movement of charged particles across the membrane from one side of the membrane to the other. And these charged particles are ions. And as these ions flow, we see these brief, very short-lived, but very dramatic changes in membrane potential. In this image, we see the various types of electrically excitable cells in the body, neurons, and then the 3 types of muscle cells, skeletal muscle, smooth muscle, and cardiac muscle. In this lecture, we're going to focus on skeletal muscle. I hope you recognize this graph. It's the graph depicting changes in membrane potential that occur during an action potential. This graph actually represents what happens in a neuron, but something very similar happens in a skeletal muscle cell. And I hope you recall that these are the phases of an action potential. As the cell moves from resting membrane potential to point B, that's the depolarization phase. Depolarization is followed by repolarization, point C. Repolarization is often followed by hyperpolarization, so now we're at point D, and then following hyperpolarization, the membrane potential is slowly restored to resting membrane potential. So here we see the same type of graph where on the vertical axis we have membrane potential in millivolt, and on the horizontal axis we have time. But we're looking at the shape of action potentials for 3 different types of electrically excitable cells. So over on the right, we have the neuron. In the middle, we have a skeletal muscle cell. And on the left, we have a cardiac muscle cell. So you can see that the neuron and the skeletal muscle cell are more similar to each other in the shape that their action potential graph takes. Something a little different happens in cardiac muscle cells, and it involves calcium ions in addition to sodium and potassium. So we're focusing on skeletal muscle tissue here, and in this brief introduction to action potentials, I can basically tell you it's similar to what happens in a neuron. So let's think about muscle tissue. I hope you remember that a tissue is a group of cells that work together to perform a common function. And that common function is to generate force to produce movement. And it's easy to think of movements of our entire body. You don't need to take a physiology class to know that muscles are involved in moving your body. But muscles also involved in moving organs, like cardiac muscle tissue in the heart is responsible for moving, pumping your blood. And then there's smooth muscle tissue, lots of places in your body. Most easily, you can think of your GI tract, your digestive system, and that smooth muscle is responsible for moving, say, the contents of your stomach into your small intestine. And so muscle cells in general, they're long, thin cells, and they contract. That's their general property. They are built for contraction. And we will see in pretty great detail how muscle cells are built for contraction. And when a muscle cell contracts, it changes shape. So the relaxed muscle cell is long and thin and quite, pliable, as we'll talk about in just a little bit. But when it contracts, that long, thin muscle cell shortens. It shortens temporarily, and it becomes shorter as well as fatter or wider. And as we'll see, those muscle cells will pull on, they'll generate tension on whatever's attached to either end. So we don't see any attachments here in the two cells or the one cell that's represented in its relaxed versus contracted state. But it is important to remember that muscle tissue is attached to at least two things. And during contraction, that muscle tissue will pull on those two things, will pull them closer together. So muscles, your skeletal muscles, like your biceps brachii, your rectus abdominis, your gastrocnemius, each one of those is an organ. And I hope you remember the definition of an organ. It's two or more tissues making up a structure. So when you look at a steak, as shown here, these are slices through an organ. Presumably they came from a cow. Cow skeletal muscle is just like human skeletal muscle. I don't know if human skeletal muscle is any tastier than cow, but histologically they're very similar. So muscles as organs include obviously skeletal muscle tissue, but you can also see adipose tissue here, connective tissue, like dense irregular connective tissue. There are also blood vessels and nerves, not so obvious here, but muscles are organs because they're composed of more than two tissues. All right, so any given muscle in your body, skeletal muscle, is attached to your skeletal system. That's why we call it skeletal muscle. So it's attached to bones. And muscles are attached to bones through sort of ropes of connective tissue called tendons. Tendons join muscle to bone. And that tendon has protein fibers that run into the periosteum, this membranous sheath that covers the exterior of a bone. And that tendon has protein fibers that run into the exterior sheath covering the muscle. So in that way, that tendon can connect the bone to the muscle. And so we're going to spend a lot of time zooming in and ultimately talking about the molecular activity that happens inside a single skeletal muscle cell. So in this image, we can start with muscle, the entire organ, and then we can pull that organ apart to examine its substructures. So if we pull that organ apart, we can look at, within this muscle here, we have bundles of muscle cells right here. This is called a fascicle. Maybe you've encountered that before. Perhaps you've taken anatomy. And a fascicle is a bundle of muscle cells. And within that fascicle, we can pull out one muscle cell. So here we're looking at a portion of one muscle cell, and notice there's a plasma membrane, right? All cells are coated by a plasma membrane. And if we look inside this one muscle cell, we see it's full of these, the artist has represented them as pink. We see that this muscle cell is full of these pink cylinders, these rods. And these rods are called myofibrils. And inside these myofibrils, we find what are known as thick and thin filaments. And those are proteins, and they interact with each other to produce muscle contraction. So I'm giving you a taste of what is to come. Ultimately, we will get down to the physiology of muscle contraction by examining what happens at the biochemical level inside the muscle cell. Skeletal muscles are able to generate body movements because they're attached to our skeleton, and most are attached to our skeleton at least two places. So we'll use this biceps brachii as the example muscle. The biceps brachii is attached to your scapula, and this point of attachment, there are actually 2 points of attachment. Your biceps brachii is attached to your scapula, your shoulder blade, in 2 places. Those are called the origins. And then if we follow that muscle down, we see another tendon down here attaching the biceps brachii to your radius. And that point of attachment is called the insertion. And when a muscle contracts, it shortens and it pulls on its sites of attachment. So when the bicep brachii contracts, it's going to generate tension that will pull the radius toward the scapula, as indicated by this arrow here. And maybe you remember, because you've seen it in another class, that the body movement produced during the contraction of a particular muscle is called the action. So in a typical anatomy class, students learn the origin, insertion, and action for several skeletal muscles. We are not doing that here in physiology, but we are going to talk about how muscle contraction happens and how body movements happen. So we're looking at an entire organ, the biceps brachii, and I've just explained to you that it's attached to two different bones. It's attached to your scapula and it's attached to your radius. And there is a hinge here in your skeletal system. Your elbow joint here acts as a pivot point or a fulcrum. And when that skeletal muscle contracts and pulls up on the radius, that contraction occurs in the opposite direction of a load. So the load, let's imagine that this blue box in the hand is some type of weight, and the load resists the movement of the skeletal muscle. So the effort, or the amount of tension that's generated, we can think of it that way, the amount of tension that's generated by the muscle, in order to move the load, the muscle needs to generate enough tension to overcome the load. Here we're going to talk about some more properties of muscle tissue. I've already introduced you to the first concept, that muscle tissue is electrically excitable because those cells can conduct action potentials. And I've already introduced you to the second concept, that muscle tissue is contractile. That means that the cells temporarily shorten, and when all the cells in a muscle temporarily shorten, the whole muscle shortens to produce body movement. And then number 3 and number 4 are new to us. Extensible means that something can stretch without tearing. So perhaps during the pandemic, you've been wearing a lot of pants that have elastic waistbands. Those are extensible waistbands, whereas paper, for example, is not particularly extensible. It's not stretchy. If you try to pull on a piece of paper, it will tear. But muscles are extensible and elastic. Elasticity is a physical property where, following stretching, a substance will return to its normal length. So let's take a closer look at what it means to be extensible and what it means to be elastic. Extensibility. Every time you stretch a muscle, you pull the insertion away from the origin. So now we're thinking about the quadriceps. And the quadriceps attach, they originate up here on the bones of the pelvis. They're attached to those bones. And the quadriceps actually insert down here on the tibia. So the origin is on the pelvis and the insertion is on the tibia. And this man is stretching his quadriceps right now because he's pulling the insertion away from the origin. So this muscle is actually getting longer. It's being extended. It can stretch without tearing. And elasticity, following stretching, the muscles return to their original length, much like this rubber band. We can stretch a rubber band, and then when we let it go, the rubber band goes back to its original length. And then this woman, she's stretching many of the muscles that occur on the anterior side of her body, on the front side of her body. So she's stretching her rectus abdominis, for example. And when she comes out of that beautiful pose, her rectus abdominis will return to its normal length. Imagine what would happen if the rectus abdominis just stayed stretched out. That doesn't happen because muscles exhibit elasticity. So our muscles, these organs, communicate with our central nervous system. Your central nervous system consists of your brain and your spinal cord. And muscles both send information to the CNS and they receive information from the CNS. And this constant communication back and forth between your muscles and your central nervous system allows your body to move in a very coordinated, intentional fashion. So I hope you recognize the vocabulary words there. We've seen them before. Afferent or afferent signals travel toward the central nervous system, and efferent signals travel away from the central nervous system. And here, in this case, we're talking about skeletal muscle. So afferent signals arise in the muscle and then travel toward the brain or spinal cord. And efferent signals arise in the brain or spinal cord and travel to the muscle. So what purpose do these signals serve? So the afferent signals, the messages arising in the muscle that get sent to the brain and spinal cord, those signals are generated by proprioceptors. Proprioceptors includes a variety of mechanoreceptors, these mechanically sensitive cells that generate action potentials, and those action potentials travel along neurons, sensory neurons or afferent neurons. And these proprioceptors are constantly generating action potentials, and the central nervous system interprets those action potentials in a way that provides information about what our bodies are doing in space. So for example, you can close your eyes, go ahead and close your eyes, and you know exactly the position of your body right now. You don't need to look to see if your hands are folded together. You don't need to look at your feet to see if your legs are crossed. You have a sense of proprioception. Proprioception also provides information about muscle tension. So your central nervous system knows what your muscles are doing. Your central nervous system knows the position of your body in space. And these proprioceptors, they provide feedback during body movements so that your central nervous system can actually, and I'll put air quotes around this, so your central nervous system knows what your body's doing. So your central nervous system can constantly adjust the efferent signals that it's sending because it's getting this constant feedback via proprioceptors in the afferent pathway regarding what's happening now. So let's think about a practical example. So your sweetie or your grandma or your roommate comes home from the grocery store, and you, being the kind and helpful person that you are, you go and you help them carry in the groceries from the store. And we don't know what's in any of these bags, and you don't need to look in a bag and make a decision about how much force you're going to need to pick up that bag. There's no need. You can be chattering away and just grab a grocery bag, pick it up mindlessly, and walk away. It might be that there's bottled water in one of these bags, and so it's going to be heavier. Or it might be that there's just one roll of paper towels in one of these bags, and so it's going to be super light. And your muscles will need to generate the appropriate amount of force to pick up either the bag containing bottled water or the bag containing paper towels. And notice or think about you actually carrying out this behavior. You do it mindlessly. It happens seemingly automatically, because as soon as you grab one of those bags, those proprioceptors are sending information about the amount of tension, the load that's in your hand. And then in response, your central nervous system is sending signals to the muscle that tell the muscle how much force to generate in order to lift that heavy bag or that light bag. So there's this constant back and forth communication between your central nervous system and your muscles. Here we see a variety of different types of proprioceptors. I'm not asking you to learn about any individual type. We're just going to talk about them collectively. But these are mechanically sensitive cells. So they respond to things like pressure and stretch. And some types of proprioceptors wrap around muscle cells so that they can tell the degree of stretch, and other proprioceptors are sort of embedded in between muscle cells so that they can get a sense of tension. Okay, so proprioceptors generate afferent signals. The central nervous system generates efferent signals. And these are motor neurons. Motor neurons deliver signals that arise in the central nervous system to muscles or also glands, but we're talking about muscles here. So the motor neurons carry the efferent signals, and as we'll see, those efferent signals cause or induce muscle contraction, and we'll come to understand that pretty well. So in this image, we can see a segment of the spinal cord. We can see some motor neurons that are leaving the spinal cord, and they come to innervate or communicate with individual muscle cells. So on the right side of that image, we see 1, 2, 3, 4, 5 individual muscle cells. Muscle cells are also known as muscle fibers. A fiber is a cell, a whole cell, with a plasma membrane and organelles and nuclei and all of that. So here we're looking at two different motor neurons. There's the green motor neuron and the purple motor neuron. And it looks like the green motor neuron communicates with 3 of the 5 muscle cells, and the purple motor neuron communicates with 2 of the 5 muscle cells. And so these 2 motor neurons control these 5 muscle cells collectively, but it's possible for the green motor neuron to send a signal and the purple motor neuron to not send a signal. So when the green motor neuron sends an action potential, those 3 muscle cells that the green motor neuron communicates with will contract. Meanwhile, the purple motor neuron is not sending an action potential, and that means that the 2 muscle cells it communicates with will not contract. Those 2 muscle cells will stay relaxed. And so in this way, our central nervous systems have a great degree of control over, for a given skeletal muscle, like your biceps brachii, for a given skeletal muscle, which cells are contracting and which are not. And that point of communication between the motor neuron and a muscle cell is called the neuromuscular junction. That's a good word. It's where a neuron meets a muscle cell. Here's what a neuromuscular junction looks like using a light microscope. So in this slide, this microscope slide, we see a portion of, I'm just going to count the number of those sort of horizontal pink lines. We see a portion of, let's see, 1, 2, 3, 4, 5, we'll count this one down here, 6. Each one of those is part of a skeletal muscle cell. Skeletal muscle cells are really long, so we couldn't fit them in our entire field of view when looking through the microscope. So we see a bunch of skeletal muscle cells here, and then this sort of spider web looking material. These are axon collaterals and axon terminals. So these are branches of a motor neuron, and right here, this is where the communication takes place. So this motor neuron communicates with this particular muscle cell at this neuromuscular junction. I love scanning electron microscopy because it really gives you a sense of how things look in 3 dimensions. So in panel A on the left, we see a portion of 3 different skeletal muscles, and we can see the neuromuscular junction here. So this is where 2 different cell types come together. This is motor neuron here. This is the axon terminal, and maybe you know the term synaptic end bulb. You will, definitely after today. And so this is where signals are transmitted from a neuron, that's one type of cell, to a muscle cell, that's a different type of cell. And this is the neuromuscular junction. And in panel B, we have an artistic representation of what's happening, where in yellow here, this is motor neuron, and an action potential travels along its plasma membrane into what's known as the synaptic end bulb. This will have various names depending on what textbook you're looking at. It's called the synaptic end bulb. And then let me highlight the plasma membrane of the neuron. This is all neuron plasma membrane. And at the neuromuscular junction, we necessarily are going to encounter also a skeletal muscle cell. So in this light blue color, and it's labeled sarcolemma. Sarcolemma is the term for the plasma membrane of a skeletal muscle cell, sarcolemma. I'll try to use both terms. It's the plasma membrane, it's the sarcolemma, same. So notice these two membranes aren't touching. The motor neuron up top in sort of like a peachy tan, that motor neuron is not physically connected to the skeletal muscle cell. And there's a little space in between these two cells called the synaptic cleft. Sometimes it's called the synaptic gap. And so this is the site of communication. A signal is sent from the motor neuron and it's received by the skeletal muscle cell. And we will understand in pretty good detail how that communication happens. And so now we're building on things that we've learned in previous lectures, particularly in the cell signaling lecture. Here's a different artistic representation of pretty much the same thing. We have a neuromuscular junction represented here, and the arrow that's traveling along the axon terminal. An axon terminal is a branch of an axon. It's part of a motor neuron, and the axon terminal leads to this bulbous region called the synaptic end bulb. So as an action potential travels along the motor neuron and reaches the synaptic end bulb, that action potential will cause these vesicles. So all of these spherical things inside the synaptic end bulb, these are all vesicles. They're sacs, they're membrane-coated sacs full of a neurotransmitter, full of a signaling molecule called acetylcholine. And with the arrival of the action potential in the synaptic end bulb, these vesicles will undergo exocytosis. And so we see acetylcholine being released, it's being secreted into the synaptic gap or the synaptic cleft. And so here in the cleft, we see all these little molecules of acetylcholine. All right, so it's a signaling molecule. It needs to have a receptor, right? The target cell needs to have a receptor to which the signaling molecule will bind. And so in the plasma membrane of the muscle cell, again, that's called the sarcolemma, we have acetylcholine receptors. Surprise! No, you're not surprised by that at all. So these little lavender or periwinkle molecules embedded in the plasma membrane of the skeletal muscle cell are acetylcholine receptors. They're proteins. They have a particular three-dimensional shape that allows acetylcholine to bind to them. And they are ligand-gated ion channels. So acetylcholine will bind to the acetylcholine receptor. The acetylcholine receptor will open up. It's an ion channel. The gate opens and it allows sodium to enter into the skeletal muscle cell. Sodium's going to move in. How do you think the membrane potential of the muscle cell will change with this influx of positively charged particles? So I've written out the steps just so you have them. These are the steps involved in cell-to-cell communication at the neuromuscular junction. This is a different artistic representation. I think it's always good to take multiple views of a particular phenomenon. So we see the same two cells represented here. We see a part of a motor neuron and a portion of a skeletal muscle cell. We have a little extra detail added here, so we can add on another layer of understanding, two other facts I want to present to you. One happens in the motor neuron and one happens in the skeletal muscle cell, or it's better represented here. So in the motor neuron, look at this. Our next layer of understanding is to talk about voltage-gated calcium ion channels. These voltage-gated calcium ion channels are embedded in the plasma membrane of the motor neuron, but only the synaptic end bulb. Like up here, we don't find any. There's no voltage-gated calcium ion channels here. They're located here in the plasma membrane around the synaptic end bulb. So they're voltage gated, which means for this protein, its particular shape is determined by membrane potential. And membrane potential is going to change dramatically. When this motor neuron is at resting membrane potential, when it's at negative 70 millivolts, these voltage gated calcium ion channels are closed. They're closed. So calcium cannot get into the cell. But with the arrival of this action potential, the membrane potential of the synaptic end bulb is going to change, right? As sodium flows in during depolarization and potassium is going to flow out during repolarization. As that membrane potential changes, this voltage-gated ion channel is going to open up, and it's a calcium ion channel. And it turns out that according to calcium's electrochemical gradient, when that channel opens, calcium will enter the cell. And not well represented here, but we'll look at it in detail on perhaps the next slide very soon. These calcium ions that flow into the synaptic end bulb, they will engage with the cytoskeleton of the cell and cause exocytosis. So the influx of calcium leads to the secretion of acetylcholine. More on that in a little bit. Let's stick with this slide for now. So we can see over here, moving on to the skeletal muscle cell, we can see in this portion of the drawing of the diagram that this muscle cell exhibits membrane potential, right? All cells in our body exhibit membrane potential. As shown here, this muscle cell is exhibiting resting membrane potential. The cell is there's a buildup of negative charges on the inside of the plasma membrane, and just along the outside of the plasma membrane, there's a buildup of positive charges, right? This is a membrane potential. And we can also see these molecules of acetylcholine binding to an acetylcholine receptor, and that acetylcholine receptor is a ligand-gated sodium ion channel. So when acetylcholine binds to the receptor opens up, the gate opens, and this allows sodium to flow along its electrochemical gradient. And as shown here, sodium flows into the cell, and sodium is a positively charged particle. So again, I'll pose the question, when sodium flows into a muscle cell at the neuromuscular junction, how is the membrane potential of the muscle cell affected? So if we think about this scale in millivolts, right, we've seen this millivolt scale before, and we'll start here. Let's say, just because it's a number we're familiar with, let's say that the muscle cell's resting membrane potential is negative 70, and we'll put 0 here. When sodium flows into the muscle cell, what happens to membrane potential? Does membrane potential get more negative? Does the inside of the cell become more negative? Or does membrane potential become less negative? The inside of the membrane becomes less negative and more positive. Do you have an answer? The answer is this one. When sodium flows into the muscle cell at the neuromuscular junction, there's a local depolarization. The inside of the cell becomes less negative because there's this influx of positive particles, positively charged sodium ions. In this slide, we're back inside the synaptic end bulb, which is part of the motor neuron. So we're going back to the motor neuron. This is, let me get the pen out. This is, right here, this is the plasma membrane of the motor neuron. And this is a vesicle that's full of molecules of acetylcholine, which is a neurotransmitter. when calcium enters the synaptic end bulb, because those voltage-gated calcium ion channels open up, the influx of calcium causes these what are known as snare proteins, or the snare complex, causes changes in these snare proteins. And you can see over here that they all have different names. Please don't know them unless you want to know them. I won't ask you about them. But calcium ions interact with these snare proteins, and these snare proteins physically pull on the vesicle and induce exocytosis. So here we can see individual molecules of acetylcholine being released from or secreted from the motor neuron. So here we are back at our neuromuscular junction. What we were just talking about happens right here in the synaptic end bulb. It's not shown here, but we know that calcium has entered the synaptic end bulb. The snare complex has pulled these vesicles toward the plasma membrane, and exocytosis is happening. And then this is the plasma membrane of the muscle cell. And so we are definitely looking at the neuromuscular junction, right, where a motor neuron meets a skeletal muscle cell. What else do we recognize here? We recognize acetylcholine receptors that are part of the plasma membrane of the skeletal muscle cell. We recognize that these acetylcholine receptors bind to acetylcholine. So it takes 2 acetylcholine molecules to bind to 1 acetylcholine receptor in order to open the receptor, in order to open the ligand-gated ion channel. So we see 2 of these red molecules of acetylcholine bound to 1 acetylcholine receptor, and that receptor changes shape. It opens up and it allows sodium ions to enter the cell. So again, the question is, what happens to the membrane potential in the muscle cell with this influx of sodium? So if now we draw the graph like this, where this is time down here, and this is membrane potential in millivolts up here, the muscle cell's at rest, and then all of a sudden there's an influx of sodium. What happens to the line next? Does it depolarize or hyperpolarize? In the previous slide, or a recent slide, we all decided, I told you, but I know you knew this, that with the influx of sodium, the membrane will start to depolarize. Membrane potential will become less negative on the inside of the cell. And so we go from -70 toward -60 toward, oh my goodness, -55. What's significant about that? What happens at -55? It's the threshold, right? So not shown in this picture, but right next door are voltage-gated sodium ion channels. And so those voltage-gated sodium ion channels, when the membrane surrounding them hits -55, they all fly open, and guess what? I think the artist is trying to represent that we end up with an action potential in a skeletal muscle cell. This is a great video. It's definitely worth pausing this lecture video and watching this YouTube video. It's fantastic. It's 3.5 minutes long. It is in Canvas. In case the video doesn't play here, it is in Canvas, and it's labeled similarly. It says cell signaling at the neuromuscular junction, 3.5 minutes. I encourage you to watch that now and come back here. There's a lot of vocabulary that goes along with muscle cells, and sometimes there are many synonyms, 2 or 3 different words that all have the same meaning, and I try to use all those words so that in the future, if you encounter those words, you'll recognize them and you're not fixed on just one set of vocabulary words. So muscle cells are also called muscle fibers, and that can be a confusing term because sometimes we use the word fiber to refer to a protein, right? Like collagen is a fibrous protein, this long stringy protein. And proteins are not cells, right? Proteins are molecules, and cells are cells. So keep in mind that a muscle cell is also known as a muscle fiber. And here's how we're going to zoom in, and I don't mean the video conference platform. I mean zoom in to go from more of a macroscopic to a microscopic view of what's happening with skeletal muscle cells. So we'll talk about the basic structure of a single muscle cell, a single muscle fiber, and we'll look inside that muscle cell to examine the arrangement of proteins in those myofibrils. And then we'll look at those individual proteins to see how they interact with each other to cause muscle contraction. And how they cause muscle contraction is known as the sliding filament mechanism. I hope you remember the phrase form follows function. It's a very useful phrase in biology, and in order to understand the function, right, and that's our focus in physiology, to understand, well, how does this happen? We often need to understand the form. What is the structure made of first, and then we can better understand how it functions. So let me give you an overview of the anatomy of a skeletal muscle cell. They have a pretty unique anatomy. There are structures in a skeletal muscle cell that don't really exist in other cell types. So let's start with what you know. What you know is all cells have a plasma membrane, and it's represented in this light blue color. In muscle cells, that plasma membrane is often called the sarcolemma because everyone needs more vocabulary in their life. That's a joke. I'll try to use the two terms interchangeably. So you know what a plasma membrane is, and I'll talk more about that plasma membrane in a moment. The interior, the volume, the stuff inside, the space inside of a skeletal muscle cell is largely filled with these pink, and they're not literally pink, but the artist is representing them as pink, filled with these pink cylinders called myofibrils. And in these myofibrils, all of these little dots that the artist is showing here in cross section, these little dots are rods of proteins called thick and thin filaments. And so we'll go there in a little bit. So so far we have a plasma membrane, we have myofibrils. What else? Here's mitochondria. Muscle cells have lots and lots of mitochondria because they have a very high demand for ATP. What else? Nuclei. Muscle cells are multinucleated, which means that in a given cell there are multiple nuclei. And this is a product of how muscle cells are formed during embryonic development. Multiple cells will fuse together to form a muscle cell, and those nuclei are kept on and kept active. A whole lot of protein metabolism happens in our cells, in our muscle cells. Our muscle cells are constantly building new proteins and constantly breaking down old proteins as a way of maintenance, of upkeep. And I hope you remember that protein synthesis requires nuclear DNA. And that DNA is the start of the process of transcription, right? Transcription and translation are two processes that occur in protein synthesis. So in having these multiple nuclei, we have multiple sets of nuclear DNA, and it makes transcription it makes transcription happen faster because we have multiple nuclei, in a sense. All right, what else can we know here? All right, let me go back to the plasma membrane, also known as the sarcolemma. And the plasma membrane of a skeletal muscle cell has this really unique feature called T-tubules. So here's a T-tubule. T-tubules, that stands for transverse tubules. They are sections of plasma membrane that tunnel in to the interior of the cell. I think about them sort of like manholes, like in the street. You're walking along the street, and then there's this hole that tunnels into, I don't know, the interior of the city. So let's imagine you're walking along the plasma membrane, and then, oh, here's a manhole, and you can tunnel into the interior of the cell. And the purpose of these T-tubules is to conduct that action potential, that electrical current, to the interior of the cell, the insides of the cell, because that change in membrane potential is important in causing the events that bring about muscle contraction. So next I want to introduce you to, and let me change colors or change widths of my pen here. I want to introduce you to, do you see this sort of tan orange web-like material here? That's known as the sarcoplasmic reticulum. And maybe you kind of recognize that word because it sounds like endoplasmic reticulum. It's very similar. It's modified endoplasmic reticulum. But of course, we're in a muscle cell. So often things start with sarco. It's the sarcoplasmic reticulum, and it has a unique function in muscle cells. It stores calcium ions. So it's like this really intricately shaped bag of calcium ions. So there's a lumen or a space inside the sarcoplasmic reticulum where there's a super duper high concentration of calcium ions. And it has this intricate shape. Well, why isn't it just a vesicle? Why does it have this intricate shape? Form follows function, right? Look at how the sarcoplasmic reticulum surrounds a myofibril. So each one of these sort of bars here is part of the I'll just call it the SR, the sarcoplasmic reticulum. So sarcoplasmic reticulum is surrounding each of these myofibrils. And that's important because at the onset, at the very beginning of muscle contraction, the sarcoplasmic reticulum becomes very permeable to calcium. And so calcium is going to diffuse out of the sarcoplasmic reticulum and into the cytosol. And that calcium will then interact with these filaments that make up the myofibril. And guess what? That's going to, and I'll put air quotes around this, turn on muscle contraction. So calcium is key in muscle contraction, and calcium is stored in the sarcoplasmic reticulum. At the onset of contraction, the sarcoplasmic reticulum releases calcium into the cytosol. What else important do we see here? Yeah, we see an important physical relationship between T-tubules and the sarcoplasmic reticulum. So let me move down here and highlight what's known as a triad. Maybe that word is familiar to you from another course. So tri means three, right? And so what we're looking at is the sarcoplasmic reticulum on this side of the T-tubule, and then we have the T-tubule, and then we have the sarcoplasmic reticulum on this side of the T-tubule. And why is that physical relationship so important? Why is a T-tubule surrounded on all sides by sarcoplasmic reticulum, by the terminal cisternae, if you remember that term? And if you didn't, if you've never heard of that before, it's okay. A triad is one T-tubule plus the two terminal cisternae of the sarcoplasmic reticulum on either side. So point being, a T-tubule is surrounded by sarcoplasmic reticulum. They have that close physical relationship because the action potential that travels through the T-tubule, that change in membrane potential, will cause calcium release channels to open up in the sarcoplasmic reticulum. That change in membrane potential will cause calcium to leave the SR and enter the cytosol. And then calcium is the key to beginning actual contraction. And we'll talk a lot more about that. One more thing on this slide. So if we pull out one of these myofibrils, right, this pink cylinder is a myofibril. If we pull it out and then we cut it in, we section it longitudinally, what do we see inside? We see what are known as thick and thin filaments, and they have very simple and appropriate names. Thick filaments are thick and thin filaments are thin, and these protein filaments are arranged into this very regular overlapping pattern called a sarcomere, and we will talk a lot about that. I like this diagram because we can see how the thick and thin filaments are arranged when we look in longitudinal section, and we can also see how they're arranged when we look at the cross section here. It seems like every thick filament, every bigger dot, is surrounded by little dots. Every thick filament is surrounded by thin filaments, and we'll talk more about that. All right, I just wrote out some vocabulary so you have it. I've covered all of these in a previous slide. And again, more explanations of the vocabulary that we just talked about. And again, explanations of the vocabulary we just talked about and their physical relationships to each other. And we're back to a very similar slide. So you can use this slide to review the structural relationships. And you can also start to think about some of the functions. What's the function of the nucleus? What's the function of the mitochondria? What does the sarcolemma do, also known as the plasma membrane? What's the purpose of T-tubules? What's the purpose of the sarcoplasmic reticulum? And why does the sarcoplasmic reticulum abut or is up against? Why is the sarcoplasmic reticulum up against a T-tubule? Why did the two things touch? Yeah, so we're going to keep putting all of those ideas together. Let's take a look at a myofibril. So those pink cylinders that we've been looking at are called myofibrils. And again, they're made up of thick and thin filaments. So now we're getting into the molecular level. So the arrangement of thick and thin filaments are shown here. The thick filaments are shown in pink and the thin filaments are shown in yellow. And then there are also a bunch of other proteins, structural proteins, that hold the thick and thin filaments together. And the arrangement of thick and thin filaments, this whole shebang right here, is a portion of what's known as a sarcomere. So let's take a closer look at what thick filaments are made out of and what thin filaments are made out of. And we'll also take a closer look at this pattern, this overlapping arranged pattern called the sarcomere. So thick filaments are made out of several hundred molecules of the same protein, and that protein is called myosin. So you can think of a thick filament as a bundle of myosin, and one molecule of myosin has a tail and two heads. And so it sort of looks like two golf clubs where their handles are wrapped around each other, or two hockey sticks where the handles are wrapped around each other. And then they're bundled together into a thick filament. And it is important to note that the tails all face the interior of the filament and the heads all stick out. So it's sort of like a bottle brush. And these heads are going to play an active role in muscle contraction. They will physically bind to, they will physically connect with the thin filaments. Thin filaments are made out of three different proteins. Actin, which is represented by the yellow balls, tropomyosin, which is represented by what looks to me like whole wheat spaghetti, and then troponin or troponin, depending on who you ask. Troponin looks like gray eggs. So these three proteins are woven together to form a thin filament. And an important component of thin filaments is the relationship between tropomyosin and actin. So here, notice that the tropomyosin is covering up that little brown dot on the actin molecule. That little brown dot is part of the actin molecule, and it's known as the myosin binding site. So all of these are proteins. So they all have a particular three-dimensional shape. Actin has part of the molecule that is specifically shaped to allow a myosin head to bind to it. And when the thin filament is in this configuration, the tropomyosin is covering the myosin binding site. So in this configuration, the myosin heads can't possibly bind to actin. There's a barrier in the way. The tropomyosin is blocking access to the myosin binding site. But at the onset of contraction, guess what? Calcium is going to bind to troponin right here. And the troponin and the tropomyosin will rotate. So these two proteins work together. And when the tropomyosin moves, it will rotate around, it'll twist a little bit. And all of a sudden, all of these myosin binding sites will be revealed, they'll be exposed, allowing the myosin heads to have access to the actin. The myosin heads will physically attach to the actin temporarily. We'll talk more about that. All right, so here we can see in physical proximity to each other a portion of one thick filament in pink and a portion of one thin filament in gray, brown, and yellow. And those myosin binding sites are not currently exposed. There's no, and I'm going to switch to purple because I believe that that's the color that is used to represent calcium in future slides. There is no calcium bound to troponin, but if there were, and I'm drawing a calcium ion, this is like the world's largest calcium ion. It's not to scale for sure. So calcium will bind to troponin, and that will cause this tropomyosin to rotate. And then all of these little brown dots will be exposed. And that means that these myosin heads are going to bind to actin temporarily. All right, so let's talk about the 3 categories of proteins that make up a myofibril. To understand the variety of proteins shown here, so some of them are in yellow, in pink, in a turquoise color, in like a light blue, and in a gray. Those are all different types of proteins. To understand these proteins better, we make categories for them, right? And that's how scientists, that's how people try to understand the world. When we see something complex, we put these complex pieces into boxes or categories, and sometimes those categories are useful, and sometimes they're not useful. I think these categories are useful to help you understand. So these proteins, this collection of proteins that make up a myofibril, we can sort them into these 3 categories: contractile, regulatory, and structural. The contractile proteins will be the actin and the myosin. Those 2 proteins are going to interact to actually make the myofibril get shorter, and that's the crux. That's the most important part of muscle contraction. And then we have regulatory proteins. This isn't the best way for visualizing that troponin and the tropomyosin that we find as part of the thin filament, right? This is not the best way to see it because they're not really visible here. But troponin and tropomyosin are regulatory proteins because they rotate, and in their rotation, they turn on and turn off muscle contraction by providing access or blocking the myosin binding site. And then the other proteins shown here, like this thing here, this zigzaggy line, this spirally thing here, for example, this turquoise chain right here. These proteins, they're not contractile, they're not regulatory, but they're structural. They hold the other proteins together to form a sarcomere and to form a myofibril. So the structure of a sarcomere. So we're looking at a portion of one myofibril, and one myofibril consists of one sarcomere after another. We can go the entire length of a myofibril and do sarcomere, sarcomere, sarcomere from one end of a myofibril to the next. So a sarcomere is defined as the portion of a myofibril that goes from what's known as a Z disc to the next Z disc. So in between these 2 Z discs, or Z lines, sometimes they're called, it's easy for me to remember because it zigzags. Maybe that helps you. And then, so we're looking at a portion of one sarcomere. This is the beginning of the next sarcomere, and we don't see this entire sarcomere. So over here somewhere, not shown, over here would be like the next Z disc. And then the same thing on this side. This is the neighboring sarcomere over here, and not shown here, but somewhere over here, we'd find the next Z disc. So let's focus on this sarcomere here. And we've blown up just a few proteins that make up that sarcomere, so it's easier for us to see. So we have our Z disc here, and then we go to our other Z disc on the other side. And hopefully you recognize thick filaments and thin filaments. The thin filaments are represented in yellow, and the thick filaments are represented in pink. And Z-discs, here's a question for you. The proteins that make up the Z-discs, are those proteins contractile, are they regulatory, or are they structural? They're structural. So the Z-disc proteins, you can see, they hold the thin filaments together, and they also hold, this is another protein called titin. You don't really need to know that. But you can see that that Z-disc helps hold those proteins together. All right, so we have the Z-discs, we have the thick and thin filaments. Other features of the sarcomere that are important for you to know, the M line. M is for middle. And this is proteins that join thick filaments in a sarcomere. And then another structure that's important to know is the H zone. The H zone is the portion of a sarcomere where there are only thick filaments and no thin filaments. So, right, we see the borders are right here. And this is certainly not a complete coverage of the structure of a sarcomere. There are other structures that we could name here, but these are the most important structures for understanding the physiology of muscle contraction. During muscle contraction, these myosin heads are going to physically pull on the thin filaments. The thin filaments are going to move toward the M line from both directions. When those thin filaments move toward the M line, the width of the H zone will get narrower and narrower. Guess what? These Z discs also move toward the M line. Everything gets pulled closer to the M line. So the whole sarcomere will get shorter in length. And this is going to happen the entire length of the myofibril. So the entire myofibril is going to get shorter. Okay, this is an H zone in a relaxed sarcomere. Relaxed means that the muscle is not contracting. There's no action potential in the plasma membrane of the cell. Calcium is stuck inside the sarcoplasmic reticulum. There's very little calcium in the cytosol, and there's no calcium to interact with the thin filaments that make up this myofibril. In the absence of calcium, we can't really see it here, but we'll take a closer look in a moment. In the absence of calcium, that tropomyosin, that whole wheat spaghetti, is blocking access of the myosin heads to the actin. The myosin binding sites are covered up. And so the myosin heads can't bind to actin. There's nothing to pull on the thin filaments. So the thin filaments are in their resting position, and here's the width of the H zone. And this muscle cell, if we can imagine that this is inside a muscle cell, all of these myofibrils are long and relaxed. That will change when calcium is released from the sarcoplasmic reticulum. The H zone will get narrower. The entire myofibril will shorten. So here we are at the sliding filament mechanism. This is how old I am. When I was in high school, in our textbook, it was called the sliding filament theory. And you know that word theory, I think it carries a little too much weight. But now most textbooks call it the sliding filament mechanism. And back in the 50s, really, so I'm actually not that old, but back in the 50s, there were competing hypotheses for, well, how do the proteins interact inside a muscle cell to produce contraction? There were competing hypotheses, and according to one hypothesis, the proteins shortened really quickly. The proteins got shorter, and then the proteins got longer really quickly. And that hypothesis was proposed by a prominent researcher and his team. And a lesser known research group proposed the sliding filament mechanism. And it was overlooked, it was ignored, it was poo-pooed for a long time until there was overwhelming evidence to show that this lesser known research team was correct. Their hypothesis was supported by the evidence. And now, of course, it's in every physiology textbook. So according to the sliding filament mechanism, the thick and thin filaments slide past each other, and this is responsible for shortening the sarcomere and of course shortening the entire myofibril. And one muscle cell is full of myofibrils. And when all those myofibrils get shorter, the whole cell gets shorter. And we call that contraction of a muscle fiber. So in this image, we have much prettier arrows indicating the direction in which the thin filaments move during muscle contraction. And that will cause the whole sarcomere to shorten. In this image, we can do a side-by-side comparison of an artistic representation on the left with what we see using microscopy on the right. And we see three panels here. Panel A is relaxed muscle, panel B is partially contracted muscle, and panel C is maximally contracted muscle. So if you just, let's think about your biceps brachii for a moment, and if you just relax your arms at your sides and let your arms just sort of hang there, that's relaxed bicep brachii. And then if you use your bicep to flex your arm at the elbow, so just like in the pictures that we saw earlier, if you bend your elbow so that you're raising your hand towards your shoulder, and if you're doing that while either carrying no load or just picking up a pen or something light, that represents the partially contracted muscle. And now imagine you want to pick up, I don't know if you're going to pick up, you want to pick up a 50-pound weight in your hand and move it towards your shoulder. That will land us at panel C, maximally contracted muscle. Okay, so let's look at the width of the H zone in a relaxed muscle, a partially contracted muscle, and a maximally contracted muscle. So let me go back to blue here. And here's the width of the H zone in a relaxed muscle cell. So there's a little bit of overlap between thick and thin filaments here. The H zone, I'll remind you, it's defined as the region of a sarcomere where there is no overlap between thick and thin filaments. And then the muscle starts to contract. These myosin heads are tugging on the thin filaments. The thin filaments are moving in from both sides. So the thin filaments have moved in from this direction and from this direction. And now look at the width of the H zone here. It's narrower, right? And the entire sarcomere is shortening. And then look down here at the maximally contracted muscle. There is no H zone. Those thin filaments have crossed the M line from both directions, and those thin filaments are overlapping each other. So this is a maximally shortened sarcomere, and there is no H zone. This is just a different artistic representation of what we've been talking about. I like this picture because here in the relaxed sarcomere, there's no physical connection between myosin heads and the actin. And then in this representation of a contracted sarcomere, we do see that the myosin heads are physically attaching to the actin, and we'll talk more about that. Okay, let's zoom out for a moment. Let's keep it real, keep the big picture in mind. So we have been talking about molecules inside of an individual muscle cell, and those molecules are part of the thick and thin filaments, are part of a myofibril, and they are arranged in an overlapping pattern called a sarcomere. And we can go the entire length of this one myofibril. Here's the sarcomere, here's the next sarcomere, here's the next one, and the next one, and we're just going from Z disc to Z disc and identifying each of those sarcomeres. And this myofibril is part of one muscle cell. That one muscle cell is part of a bundle of muscle cells called a fascicle. And that fascicle is part of a muscle, and that's an organ, a skeletal muscle. Here's your bicep brachii. So during muscle contraction, when those thick and thin filaments slide past each other, each sarcomere will get shorter. This sarcomere will get shorter. All of them get shorter. So the whole myofibril gets shorter. If the whole myofibril gets shorter, the whole cell gets shorter. If the whole bundle of cells gets shorter, the whole fascicle gets shorter, and all the fascicles get shorter, the whole muscle gets shorter. When the muscle gets shorter, it pulls on, let me switch colors here. When the muscle gets shorter, it pulls on its points of attachment. So remember that tendon that connects the bicep brachii to the radius and other tendons that connect the bicep brachii to the scapula? This whole muscle temporarily shortens and pulls the radius toward the scapula, and that's called the action of the muscle. It's flexion of the arm at the elbow. Okay, we're diving into the molecules, to the cells and the molecules again. We just zoomed out for a breather and we're zooming back in. And here we see a very much simplified skeletal muscle cell. It's a schematic diagram. A schematic diagram is representational in a teaching environment. It's used for learning purposes, right? Like skeletal muscle cells aren't square. That should be a clue here. So we'll take a look at the structures that are being represented here, because there are quite a few. And I'll point those out, and then we'll think about, well, what's happening here? What are the processes that the artist is trying to show us? So let me switch to, how about green? There's not a whole lot of green on this side. So what do we already know? What do we see here? This light blue line surrounding the cell, that's the plasma membrane of the muscle cell, also known as the sarcolemma. This invagination represents a T-tubule. This sac here, this whole sac, I won't surround the whole thing or else I make a big mess out of the picture. But this sac here represents the sarcoplasmic reticulum. And remember, the sarcoplasmic reticulum has a close physical relationship with the T-tubule. Remember that the sarcoplasmic reticulum is full of calcium. That's the major job of the sarcoplasmic reticulum, is to store calcium ions when the muscle cell is relaxed. And calcium is represented by those little purple dots inside. So this cell has to be relaxed because all of the calcium is inside the sarcoplasmic reticulum and none of the calcium is in the cytosol. Other features, new features of the sarcoplasmic reticulum for us. These little sideways sort of triangles, they look like sideways camping symbols to me. When you're on a road trip and you're looking for a campsite, you look for these little triangles. Here they're on their side. These black triangle-ish structures represent what are known as calcium release channels. So right now they're closed. It's a transmembrane protein in the wall of the sarcoplasmic reticulum that changes shape, and right now they're closed, but later on we'll look at what happens when they open. When they open, calcium will be allowed to diffuse, right? There's a very high concentration of calcium inside the sarcoplasmic reticulum and a very low concentration of calcium in the cytosol. So once these calcium release channels open up, calcium's going to very quickly, it's a very steep concentration gradient, so calcium's going to move fast, will leave the sarcoplasmic reticulum and enter the cytosol by diffusion. Another feature of the sarcoplasmic reticulum are these little spinning wheels. The spinning wheel is the way that the artist is representing a calcium ion pump. Now you recognize that word pump, right? Pumps perform active transport. So these calcium ion pumps are constantly using ATP to move calcium against its concentration gradient. They are pumping calcium out of the cytosol and into the sarcoplasmic reticulum, uphill or from low to high, and they are constantly pumping 24/7. All right, the next structures that I hope you recognize here, this is a myofibril, right? And there are two myofibrils. And the artist has taken care to show you that the myofibril is physically connected to the plasma membrane. And it's physically connected on the other side as well. So I hope you remember that this myofibril, during muscle contraction, it will get shorter. Each sarcomere will get shorter. And so the whole myofibril will get shorter. And that myofibril will pull on the plasma membrane to help the muscle cell physically get shorter. What else do we see here? Stuff that will become important later, but I'll point it out now. We're not going to talk about it right now. But when we get to muscle metabolism, we'll talk more about the role of mitochondria. We'll talk more about the role of glycogen and myoglobin. All All right, I think that's it for now. Let's look at the next slide. So here we can look at those thick and thin filaments and what's happening when calcium is absent from the cytosol in a relaxed muscle cell versus what's happening when calcium is present in the cytosol, when calcium has left the sarcoplasmic reticulum and diffused into the cytosol. So let's take a look at what it looks like when calcium is not present. When calcium is not present, it's not bound to troponin, so we don't see any calcium here. And this tropomyosin, the whole wheat spaghetti, is blocking the myosin binding sites on actin. The actin's not really super represented here, but the myosin heads cannot physically connect to actin. The thick filaments can't bind to the thin filaments. All right, let's move over to the panel on the right, where now you can see calcium is represented by these little purple balls, and calcium binds to troponin. When calcium binds to troponin, the tropomyosin, the whole wheat spaghetti, rotates out of the way, and now we have exposed myosin binding sites on actin. So those little brown dots are the myosin binding sites on the actin molecule. And look what happens. Dun, dun, dun. The myosin head binds to actin, and we call that a cross bridge. When the myosin head is physically connected to actin, we say that cross bridges form, or there are cross bridges exist. And when there are no cross bridges, it means that the myosin head is not bound to actin. All right, we're putting the pieces together. I hope you recognize all of the components here. So again, on the left side, we have a relaxed muscle cell. And on the right side, we have a muscle cell during contraction. So let's start with the relaxed. And let me remind you of what you know. You recognize the sarcolemma, you recognize this T-tubule, you recognize that this is the sarcoplasmic reticulum. It's full of calcium because these calcium pumps are constantly transporting calcium out of the cytosol and into the sarcoplasmic reticulum. And there's no action potential in the sarcolemma, so these calcium release channels are closed and calcium is stuck inside the SR. What else do we see? Because there's no calcium here, the tropomyosin is covering up the myosin binding sites and there are no cross bridges. So the myosin heads are not attached to actin. This H zone here is nice and wide. This is a relaxed sarcomere and the whole cell is nice and long. All right, let's move from left to right. So we'll take a look at what's happening inside the cell during muscle contraction. In order to bring about muscle contraction, an action potential travels along the plasma membrane of the muscle cell. It travels down the T-tubule, so we have this massive physiological current happening here. That change in voltage, oh look, the calcium release channels have opened up and calcium is pouring out of the sarcoplasmic reticulum and into the cytosol. When it's in the cytosol, calcium will bind to troponin, and that tropomyosin will rotate out of the way, exposing the myosin binding sites, and oh look, there are cross bridges. Cross bridges have formed, the contraction cycle has started, and that's where we're going to go next. The contraction cycle is also known as the cross bridge cycle, and it's a repeating sequence of events that cause the thick and thin filaments to slide past each other. And you're familiar with all of the players, except maybe ATP. That will be the new player in this story. So there's a lot going on in this diagram. We'll break it down. I do want to start by saying it's a cycle, so we could start anywhere in this sequence of events, and we'll end up right where we started. So I'm going to start with number 1, just because that's what the artist had in mind, right? But before I start with number 1, I want to introduce you to the new players. Or no, let's talk about the old players. What do you already know? What do you already know? You already know that this is a thick filament. You already know that this is a myosin head. You already know that this is a thin filament. You already know that this is calcium, represented by those purple balls. You already know that calcium binds to troponin, and when that happens, the myosin binding sites are exposed. You already know that that allows for the formation of cross bridges. It allows the myosin head to attach to actin. That's a cross bridge. There's no cross bridge here. All right. The new things that you don't yet know, is the contraction cycle involves ATP. And ATP binds to the myosin head. The myosin head includes an ATP ACE. What does an ATP ACE do? It cleaves, or it cuts apart, it breaks apart a molecule of ATP. And what happens then? What's the significance of breaking apart ATP? It releases energy. Here, that energy is used to drive a conformational change in the myosin head. Myosin's a protein. Lots of proteins change shape as part of doing their job, and myosin is one of them. What causes the myosin head to change shape? A couple of things. ATP is one, and connecting to actin is another. So notice that this ATP is bound to the myosin head. And as we go from one step to the next, notice that ATP gets cleaved into ADP and P. Notice that the conformation, the bend in the myosin head has changed. We go from this like super bent shape to this less bent shape. That's a conformational change. And we're going to go back and forth from the super bent shape to the less bent shape. All right. I want to erase all of the drawings on this diagram so that we can go through the contraction cycle. Let's get rid of all of these. Start with a clean slate. Now that we know all the characters that are in our story, we can talk about how these characters interact. We can go through the plot, right? So let's start with step one. The myosin heads hydrolyze ATP. That just means that they cleave ATP and energy is released. And that energy drives a shape change in the myosin head. So this myosin head goes from what's sometimes called the low energy position to the high energy position. What are you doing that for? All right, so we go from the low energy position to the high energy position, and now we're at step two. The myosin heads bind to actin, forming cross bridges. Notice as we go from step one to step two, that phosphate group popped off, and that allows cross bridges to form. Here's a cross bridge. As soon as that cross bridge forms, myosin changes shape again. It goes from this high energy position, this less bent configuration, to this more bent configuration. And I wish the artist made that a little more dramatic. But as we go from step two to step three, the myosin head goes from a high energy shape to a low energy shape. And sometimes in class, I'll demonstrate throwing a baseball pitch, right, where the pitcher has, in the windup, takes the ball and places it sort of behind her head. And then as she throws the ball, she goes from a high energy position to a low energy position. That's sort of one way to think about what the myosin head is doing. So as that myosin head is going from a high energy position in step two to a low energy position in step three, it physically shoves the thin filament. And that's called a power stroke. Notice the position of this thin filament relative to the thick filament. Here they're lined up, and here they're not lined up anymore, because the myosin head has physically pushed the thin filament in this direction, and we can assume that the M line, the middle of the sarcomere, is in that direction. All right, so in going from step two to step three, we perform a power stroke. And then in going from step three to step four, we break cross bridges. how do we get the myosin head to detach, to separate from actin? The way to do that is for a new molecule of ATP to bind to the myosin head. Notice that the ATP isn't cleaved yet. It's not broken. It's still an intact molecule of ATP bound to the myosin head, but just the binding of that ATP causes this myosin head to change shape a little bit, lowering the binding affinity between myosin and actin. So when ATP binds to the myosin head in step 4, myosin has no binding affinity for actin. But when this ATP gets cleaved in moving from step 4 to step 1, energy is released, the myosin head changes shape, and now it has a high binding affinity for actin. When this phosphate pops off, the myosin will bind to actin, moving from step 1 to step 2, forming a cross bridge. As soon as the cross bridge forms, a power stroke occurs. That's moving from step 2 to step 3. And the thin filament is physically pushed toward the M line. The next step is to break the cross bridge. Moving from step 3 to step 4, ATP binds to the myosin head, and that causes the cross bridge to break. Moving from step 4 to step 1, ATP is hydrolyzed, it's split apart. That energy that's released causes a shape change in the myosin head. The myosin head goes from its low energy position to its high energy position, and we're back where we started. This happens millions of times a second. The myosin heads operate asynchronously, so they're not all in the same phase of the cycle at the same time. At any one moment in time, some of the myosin heads are bound to actin, others are not. The myosin heads all work together to shove the thin filament toward the M line. Now would be a great time to pause this lecture recording and to go watch each of these videos to really reinforce the concepts that we've just covered. These links are posted in Canvas. Okay, I'll see you in a few minutes. We have a few more topics to cover in this lecture segment, and they're all related to muscle contraction. Our next vocabulary term is excitation-contraction coupling. So in the process of science, in the scientific process of trying to understand the physiology of muscle contraction, there was a lingering question. Scientists knew that excitation of the plasma membrane, right, the action potential, was somehow linked to muscle cell shortening. So we knew that excitation and contraction were linked, but we didn't know how. We didn't know why an action potential in the plasma membrane preceded or caused contraction. And now we understand it, and we know that link between excitation of the plasma membrane and shortening of the cell. We know that link is calcium. Calcium gets released from the sarcoplasmic reticulum. It binds to troponin, and that is when the contraction cycle begins. So there's this delay, this time delay between excitation in the plasma membrane, the action potential, and the actual shortening of the cell. And that time delay is called the latent period. So there's a lot going on in this diagram, but you've seen all of the pieces before. And if it's helpful for you, use this diagram, follow the numbers to make sure you understand the whole story. I'm not going to do that with you now because I think we've been through it all. But what I do want to use this picture for is illustrating what gives rise to the latent period. And I'll show you that on a graph next. But remember, the latent period is a delay between the events that are occurring in the plasma membrane, excitation of the plasma membrane, and actual cell shortening. So if we think about what's going on up here, here's the initiation of the action potential in the plasma membrane of the muscle cell. And then we go through all of these processes here before we actually see cell shortening. So all of the events that occur between these 2 points, between, I guess it's point number 1 on this figure and point number 6 on this figure, the time it takes for those events to occur gives rise to what we observe as a latent period. We can use this graph to further our understanding of the latent period. So there are 2 graphs here. Up top, that's a familiar graph for you. It's a muscle action potential. So in the top panel on the vertical axis, we see membrane potential of the muscle cell measured in millivolts. And on the horizontal axis, we see time. And in the lower panel, on the vertical axis, we see muscle fiber tension. You can also think about that as muscle fiber length. How relaxed versus contracted, how shortened is the muscle fiber? So let's start with the top panel. Let's start with what we know. Muscle cells have a resting membrane potential of -90. So far, we've been mostly talking about neurons and using the example resting membrane potential of -70, but all the same principles apply here. So the muscle cell goes from resting membrane potential to a depolarization phase, a repolarization phase, hyperpolarization, and then back to resting membrane potential. That's all in that green line. We can overlap that with what we observe for muscle tension or muscle length. So we can see at time 0, that's the beginning of the action potential in the muscle, in the plasma membrane. But notice there's a latent period in the lower graph. At time 0, nothing's happening in terms of a change in the length of the muscle cell. The muscle cell is staying the same length. It's not contracting. It's not generating tension. And so there's that little delay called the latent period. And it's caused by, the reason the latent period exists is it's the time delay between the excitation of the muscle plasma membrane and the actual onset of the contraction cycle. That's responsible for the latent period. And what joins those 2 events is calcium. Calcium is responsible for excitation, that's the green line, the top line, contraction, the orange line, coupling. So we've worked with this graph before, and I can use it once again to illustrate the reason for the latent period. So here on the panel on the left, we have a relaxed muscle cell. Notice that there's no action potential in the plasma membrane. Notice that the calcium release channels are closed. Notice that we have a big wide H zone. And then we move over to the panel on the right, and this would be time 0 up here, the initiation of an action potential in the plasma membrane. And it takes time for all of these events to happen for the contraction cycle to bring about a shortening of the sarcomere. Following muscle contraction, a muscle will relax. And how does relaxation occur? Step 1 in relaxation is to eliminate acetylcholine from the synaptic cleft. We need to stop that cell-to-cell signaling between the motor neuron and the skeletal muscle cell. So we talked about the cessation of the signal back in our cell signaling chapter. And 2 of those processes are employed here. Number 1, acetylcholine will diffuse away from the synaptic cleft. It's a molecule, it's a substance, it diffuses from an area of high concentration to an area of low concentration. So it diffuses away from the cleft. Number 2, we have degradation. That's our second mechanism, because in the cleft, there's an enzyme present called acetylcholine esterase. And acetylcholine esterase, it's an enzyme, that ACE ending is a clue. Acetylcholine esterase, or ACHE, breaks apart. It cuts up acetylcholine. And once that acetylcholine is degraded, it can no longer bind to acetylcholine receptors. So when those acetylcholine receptors have no ligand bound to them, remember they were ligand-gated ion channels, the gate closes and no more sodium can enter the cell. And the sodium-potassium pumps will restore resting membrane potential. Number 2, inside the muscle cell, In the absence of an action potential, the calcium release channels in the wall of the sarcoplasmic reticulum are closed. Those calcium pumps, those little blue spinning wheels, are constantly using ATP to pump calcium out of the cytosol and into the sarcoplasmic reticulum. So as the concentration of calcium in the cytosol goes down, calcium diffuses away from troponin. And the tropomyosin, once again, changes shape to cover up the binding sites on actin. When troponin and tropomyosin are covering the myosin binding sites, then cross bridges become impossible. There's no physical connection between the thin filaments and the thick filaments. And so the thin filaments will then slide backwards, so to speak. They'll slide away from the M line. And that makes the H zone wider again. And the sarcomere, and of course the muscle cell, will return to its original longer length. And we call that muscle relaxation. So you're in a position to understand the biochemistry behind a phenomenon known as rigor mortis. And many people watching this lecture have experience with rigor mortis, certainly not in your own body because rigor mortis happens after a body dies. But perhaps you've experienced it in somebody else's body or in the body of a pet perhaps who has recently died. So rigor mortis is defined as muscle rigidity. So the muscles become hard, they become stiff. That sets in somewhere usually between 3 and 24 hours after death. And this is why sometimes in movies and books you hear about the dead body being referred to as a stiff. Yo, what are we going to do with that stiff? So let's understand the biology of rigor mortis. So what happens when a body dies? Death is actually, at the cellular level, death is a process. So when somebody's heart stops beating, it's not like every single cell in that person's body dies immediately. That's not true. Death comes about at the cellular level as cells use up their supply of ATP and they have no more oxygen or glucose. They have no more ingredients for making new molecules of ATP. The cardiovascular system isn't functioning anymore. There's no new delivery of oxygen and glucose. So cells run out of ATP, and that's really when individual cells die. They can't maintain homeostasis without ATP, and if there's no homeostasis, there's no life. So what happens to a muscle cell? Like any other cell, a muscle cell will use up its supply of ATP and it won't be able to generate any more. So without ATP, all of the pumps will stop. The sodium-potassium pumps will stop. Those calcium pumps in the sarcoplasmic reticulum will stop. And our membranes, all cell membranes, are leaky. They're not perfect. Is it possible for ions to leak across? Yeah, totally, between little gaps in the phospholipids. And so our membranes become leaky and there are no pumps to reverse the leaking. And so what happens is that calcium leaks out of the sarcoplasmic reticulum. You remember that there's a very steep concentration gradient for calcium, from inside the sarcoplasmic reticulum to outside the sarcoplasmic reticulum. The cytosol normally has very, very little calcium. So without those calcium pumps operating, calcium will leak out of the sarcoplasmic reticulum and into the cytosol. And once it's in the cytosol, that calcium is going to bind to troponin. Tropomyosin will move out of the way and cross bridges will form. So thick and thin filaments in all the sarcomeres of all the myofibrils of all the muscle cells will have cross bridges. Now the contraction cycle can't occur because there's no ATP to break the cross bridges. So cross bridges form, but there's no power stroke and there's no ATP available to break the cross bridges. So for many hours, the muscles feel rigid because cross bridges have formed and there's nothing to break them. rigor mortis will go away gradually as other membranes leak and protein digesting enzymes go rogue. So there are proteases inside our cells, and these are enzymes that degrade, they break apart the peptide bonds in proteins. And they're pretty indiscriminate. They'll break apart any protein they encounter. If it's a protein, if it's the right peptide bond, they'll cut it. So we have these protein digesting enzymes. They're normally secluded away for our cellular safety. These protein digesting enzymes are usually kept inside vesicles, and they're not allowed, they can't be sprinkled around the cell because it's too dangerous. They'll chop up any protein that they encounter. So now, following death, there's no way to maintain these vesicles, and the vesicles become leaky, and these protein digesting enzymes get out and they catabolize any protein that they encounter, including myosin and actin. So as the contractile proteins are digested, are cut up by protein digesting enzymes, by proteases, rigor mortis will gradually fade away because these proteins will be chopped up into amino acids, and obviously there's no more cross bridges. So I just explained this on the previous slide. Rigor mortis will end when these protein digesting enzymes indiscriminately break down proteins, including actin and myosin, and that's when the muscles become soft and pliable again. All right, I believe this is the last concept in this lecture. You are in a position to understand the mechanism of action of Botox. Most people have heard of Botox, mostly in a cosmetic context. So Botox stands for, or it's short for botulinum toxin. And a particular type of bacterium produces this toxin. And it is a toxin, it's a poison, it kills people because it blocks the release of acetylcholine at the neuromuscular junction. So it interferes with that cell-to-cell communication between the motor neuron and the muscle cell. It's a foodborne illness that leads to a horrible death if you eat food that has some of this type of bacteria in it, and that bacteria survive your digestive system and your immune system, and they are able to make this toxin. This toxin could potentially kill you by causing respiratory arrest. So that means that the diaphragm stops contracting and you stop breathing. A few decades ago, Botox, or botulinum toxin, was started to be applied in both cosmetic and therapeutic settings. So in a cosmetic setting, Botox can be injected into the small muscles in the skin. These muscles are responsible for our facial expressions, and Botox can induce temporary paralysis by disrupting communication between the neurons and the muscle cells. So parts of the face where Botox is injected become paralyzed, and some people think that makes them look better. There are also therapeutic uses, and therapeutic means there's a bona fide medical use. It improves a medical condition. So some medical conditions involve too much communication between a motor neuron and a muscle cell, or a motor neuron and a gland. And Botox can be used to disrupt that communication. So if you've ever noticed somebody, or maybe you love somebody, where both of their eyes don't point in the same direction, sometimes it's called having a lazy eye. Sometimes that condition is caused by overstimulation of one of the extrinsic eye muscles. So attached to the exterior of your eyeball are 6 skeletal muscles that are responsible for pulling your eyes and your eyeballs in different directions. So you can hold your head still and you can look up, you can look down, you can look left, you can look right. It's because these skeletal muscles are physically pulling on your eyeball to rotate it in a particular direction. Well, for some people, one of those muscles is constantly being overstimulated and pulling the eyeball in, I'll put air quotes here, in the wrong direction. So one way to correct that is to paralyze that muscle using Botox. Another example of a therapeutic use for Botox is to reduce the incidence of migraines. So certain types of migraines, these are very intense, debilitating headaches. Certain types of migraines are caused by overstimulation to certain muscles in our head. So particularly around the occipital region, so the back of the base of the skull. So by limiting communication at the neuromuscular junction, we can prevent these muscles from contracting and giving rise to this headache that can go on for days or weeks. Let's take a look at how Botox works. So this is a very simple diagram. I may have even found this on a dermatology website or a cosmetic website. So we're looking at the neuromuscular junction, and the information here is correct. It's just overly simplified for our purposes, but I'll use it to introduce the fact that Botox prevents the motor neuron from releasing acetylcholine. So in panel A, that's the normal functioning of the neuromuscular junction, and in panel B, that's the neuromuscular junction under the influence of Botox. So that motor neuron can have an action potential, but that action potential never causes exocytosis of acetylcholine. Well, how does that work, Laura? Let's use this diagram for a much more appropriate level of understanding, appropriate for our level, for class purposes. So what's going on here? So we're at the neuromuscular junction. Let's use, how about red? Because there's not a whole lot of red on this image. So look here, this is the plasma membrane of the motor neuron, and this is the plasma membrane of the skeletal muscle cell. You'll recognize pretty much everything here. This is a vesicle containing acetylcholine. This is an acetylcholine receptor. And what happens here? This is the toxin molecule. This is the poison that was produced by bacterium. It's got a sneaky little mechanism. Look at this. It sort of tricks our cells. It binds to a receptor on our cell's plasma membrane and causes our cells to perform endocytosis. So our cells get tricked into pulling this botulinum toxin inside. And then once this toxin is inside our cells, pieces of the toxin, and there are different forms of botulism toxin, and that's what types B, A, C, etc. They all, in one way or another, they all interfere with the snare complex. Remember that snare complex that we talked about earlier in this lecture? The snare complex is responsible for essentially dragging this vesicle to the plasma membrane so that exocytosis can occur. In the presence of botulism toxin, that snare complex does not form and exocytosis becomes impossible. Without acetylcholine in this synaptic space, these receptors are never going to be activated. They'll never open up and allow sodium to enter. So the central nervous system is sending the message, but the message never gets transmitted from neuron to muscle cell. All right, that brings us to the end of segment 1 of 3 segments concerning muscle tissue. And in today's lecture, in this first segment, we covered all of this. And in the next segment, we'll pick up with muscle metabolism and we'll talk about the biochemistry behind muscle fatigue. Thanks for watching.